and novel antibiotics. Can you please use the question and answer function of Zoom to ask questions after the talks? We'll take 1 or 2 questions after the first talk and then open the floor to general questions when we've heard both speakers. Our first speaker today is Jeremy Lakey. He's Professor of Structural Biochemistry at Newcastle University Medical School. He's been interested in bacterial membranes and antibiotics since his PhD days, and this interest led to the formation of 2 spin-out companies concerned with the engineering of membrane proteins. In 2002, Jeremy was awarded a BBSRC Research Development Fellowship to investigate membranes on the nanoscale. This in turn led to an interest in neutron reflectivity. And today, Jeremy is going to talk about studying the surfaces of bacteria using neutron scattering, finding new openings for antibiotics. Over to you, Jeremy.
So good morning, everybody. Share. Is that okay, John?
That's good, thank you.
Okay. Right, good morning, everybody. Thanks for the opportunity to describe some of the work that we've been doing using neutron scattering in our work on outer membrane proteins of bacteria and the relationship with the need to work on new ways of finding new activities for existing antibiotics in a way, but also finding new antibiotics as well. So just to kick off, like most biologists, I kind of started off with a fascination for biology, looking at things like insects in the garden and plants. And then when you get a microscope, you can start to see things. And one thing we do forget is the immense diversity of microbes. And whilst I'm going to be talking about the rather boring microbes in the bottom right-hand corner of this, just remember there's so much variety out there down at the microbial level. They're not just sort of bags of life without much to show for themselves. But what we are interested in is these bacteria down in the bottom right-hand corner, because they tend to be the ones that we have problems with in health. And so first of all, they can be easily differentiated amongst themselves by the surface membranes. And this goes back to the work of Hans Gram in the 19th century. And he, like a lot of biologists at the time, microbiologists at the time, he was using the newly synthesized dyes that were coming onto the market and finding ways in which living cells under the microscope reacted differently to the different dyes that you could apply to them. And he was able to divide the bacterial world into 2 groups, which eventually became called gram-positive and gram-negative by the way that they took up his gram stain. And later on, it became clear that this division was because the gram-negative bacteria, which didn't take up the gram stain, have an extra membrane on their surface. So that is the sort of design, cartoon design on the right-hand side. And this extra membrane provides them with an extra level of protection from the outside world, which the gram-positive bacteria and any other living cell doesn't have. So the gram-negative outer membrane is a unique barrier in biology. And for that reason, it's kind of interesting to study. And so here we can see some gram-positive round cells and some gram-negative rod-like cells. And so this shows the penetration of the dye into the bacteria, which is affected by this outer membrane. And the other reason we should care is that gram-negative bacteria are one of the most likely ones that you're going to pick up in a hospital. A few years ago, it was things like MRSA, but we've been able to control those quite well, and Clostridium difficile, and gram-negative bacteria really coming up on the inside track. And whilst this ought to be well known to all of us, and this increase in gram-negative bacterial resistance to antibiotics is important. Remember that people who go into hospital with COVID and end up on ventilators will probably have to be protected from in-hospital acquired infections by large doses of antibiotics. So it remains important whatever the particular pandemic we're undergoing at the time. And so, so try and get this. So here we have a movie from some colleagues of mine at Newcastle. And on the left-hand side, we have some bacteria which are being treated with an antibiotic. And we can see them popping as the cell membranes pop due to the cell wall being degraded. And on the right-hand side, we have bacteria which weren't treated with antibiotics. But these are equivalent to those which are resistant to antibiotics. And so, you know, clearly uncontrolled growth like that we see on the right-hand side is what leads to systemic infection and serious consequences. And we really want the bacteria to be like the ones on the left and susceptible to the antibiotics that we apply to them. So this is a very complicated diagram of a gram-negative bacterium, and I don't intend to go through all of this, but in some ways the complexity tells its own story, because there are many different ways in which bacteria can develop antibiotic resistance. And this is a whole series of those methods: degrading the antibiotics, pumping them out of the cell here. But one of the ways in which they can control the effect of antibiotics is simply to stop them getting in to the cell by making use, and this is a gram-negative bacteria, making use of this outer membrane here as the first line of defense to stop the antibiotics getting through. And so that makes a very tight layer there, which is impermeable. But the bacteria have to communicate with the outside world, have to get food in, have to get rid of waste products. And so they have these pore-in proteins, which make the outer membrane a molecular sieve, only allowing certain molecules through. And it's this outer membrane and its associated proteins, which is the study of my research, and where I've used neutrons over the last couple of decades, in fact. So the important thing is that this outer membrane is a molecular sieve. So the gram-positive bacteria, which I introduced to you before, with their simpler cell wall and single membrane, are targeted by these molecules on this red. So this is a plot of molecular weight against polar surface area. So the more water-soluble molecules are up this end, less water-soluble molecules are down this end, and this is simply size. So there is a general trend, a correlation between polar surface area and size, which goes up about this angle. And the gram-positive bacteria essentially have drugs which affect them right the way out to here, up to 2,000 Dalton's molecular weight, and with a really kind of an average polarity. The gram-negative bacteria, because of the cutoff size of the molecular sieve, are affected by these blue triangles. And you can see that they go up at a steeper angle. So actually these molecules are generally more polar, more water-soluble than these. And that's because they have to get through the water-filled pores of the molecular sieve. They can't get through the membrane, as I will describe in a minute. There's two outliers to this. One is a macrolide, which is a hydrophobic antibiotic, which is slightly larger than the cutoff. But the real standout one is polymyxin B, PMB, which I will describe later. And this is a molecule which directly targets the outer membrane itself. It doesn't have to cross the outer membrane to carry out its function. It actually goes straight for the outer membrane. And in disturbing that, it causes after effects. So we'll look at that in a minute. But the important thing is this red line, about 600 Daltons. So at the moment, we're limited to antibiotics less than 600 Daltons. And if we could find some way of breaking through this red line, we could actually start to use some of these molecules out here, which are actually functional, actually active against gram-negative bacteria, if only they could get across the membrane. So this is a model which was put forward by Hiroshi Nakado and Marty Vara way back in 1985, but it shows essentially a thing. The outer membrane is an asymmetric structure. On the outside are these molecules called lipopolysaccharide. On the inside is normal kind of phospholipids. This outer layer is incredibly impervious, meaning that molecules, particularly lipophilic molecules, cannot get through this layer, whereas they could easily get through the underlayer. And so this LPS is linked together by magnesium or calcium in these links here. That also links it to the proteins. And so this tight barrier here means that anything has to go through this little thing. And so this is the 600 Dalton cutoff here. So I'm briefly going to talk about antibiotic LPS interactions as studied by neutron reflection. So for those of you who haven't done neutron reflection, think of it as an extension of looking at the light which is reflected by the surface of water when it has an oil film on the surface. We get different colors from the oil film, even though the oil could actually be colorless. And this is because of the differential reflection of light either from the top of the oil-air interface or the oil-water interface. And that extra path length allows us for constructive or destructive interference. And by analyzing that, we could actually find out two things. We could actually find out the distance here, but we could also find out the difference in refractive index between the oil and the water, because that's what's causing the reflection at the interface. So if we then scale right down, we could use X-rays, but we're using neutrons here because they, for reasons I'll explain in a minute, much shorter wavelengths. We can actually look at a membrane, which is a few nanometers, 5 nanometers thick, and we can get the resolution of that. And so we can measure D, and we can measure refractive index or scattering length density of neutrons in very similar ways. So, and because different components have different scattering lengths, we can start to take this apart. So why do we use neutrons? Well, one of the reasons is that the sample is here, is held, this is the silicon surface with a membrane on it with the water in here. This is the silicon block. To actually get radiation in here to with suitable wavelengths to reflect off the surface, we need to use neutrons because they penetrate all of these sample environments. But the other really useful thing is if we look on the right-hand side, you'll see that the lipids, so the membrane is now upside down. This is how it is in the experiment. The lipids on the inner layer of the membrane, which is at the top now, are red, and those on the outside are brown. Well, the red ones are colored red because we've made those out of deuterium rather than hydrogen on all these carbon-hydrogen chains. And that is very, very useful for neutrons because neutrons are reflected differently from deuterium than they are from hydrogen. These are the sugar groups on the top of these special lipopolysaccharide lipids. Okay, so this is the experiment. The neutron beam is coming down here and reflecting off of the bottom side of this silicon surface here. So here's the result. I've turned the model around 90 degrees so it fits with the results. This is the original data. This is the model fit. And we get this biphasic behavior here because those inner tails there have a scattering length density or neutron refractive index which is very high, whereas the hydrogenous, these brown ones here, have a very low one level here. The water content, so this is 3 different experiments carried out at 3 different D2O concentrations, 0, silicon match water and 100% D2O. And we can look at the amount of water at various parts of the membrane by how this varies with the change in external solution. And so this here, we can see see the change in hydration here. So that tells us there's a lot of water here. There's no water here in the hydrogenous layer, and there's a little bit of water there. But you can see that the power of neutrons also is that we can actually see the asymmetry of this membrane. Phospholipids here, lipopolysaccharides on the outside. And that's something that in my experience, only neutron reflectivity can do in a model like this. Why is that important? Well, we can show that, for example, if we take away the stabilizing calcium ions, that we go from this strong double peak to a flattened peak. And that tells us that the asymmetry here has broken down. We take away those binding calcium ions or magnesium ions, and the whole membrane becomes symmetrical and mixes up. That confirms studies by molecular dynamics led by Simon Khalid at Southampton. So what else can we find out? Well, neutron reflection recently revealed the gel-to-liquid phase of LPS, which hadn't really been shown in a membrane situation before. And as we raise the temperature, we can see that the membrane gets thicker, but we maintain the asymmetry of the layer. So the membrane stays asymmetric, it's perfectly fine, but these things here, and this is around body temperature. This lipid was extracted from bacteria which were grown at body temperature, and we can see the change in fluidity there. And that was a matter of debate before we were able to measure it. So why is that important? Well, that polymyxin antibiotic that I showed you before, if we add it to a membrane at room temperature or below lipid phase transition, we get no binding at all. So the data before and after just superimposes on itself here and here. If we then raise the temperature to above the phase transition and add the polymyxin antibiotic, we get disruption of the surface. We get mixing of the asymmetric layer, we get loss of lipid, we get water penetration, and you can see we go towards this blue structure here, which is nothing like the nice membrane that was. So we've started, so it's quite clear that the old papers which showed that lipid polymyxin was temperature dependent is entirely dependent upon this liquid phase transition. So neutron reflection has helped us show that we have this dense hydrated polysaccharide layer which resists large hydrophobic layers. We've confirmed and strengthened the data which showing that tight interactions by calcium are essential to make this tight layer here, which resists things like antibiotics. And then the fluid bilayer on the inside occurs, and we know that fluid bilayers are much more resistant to water-soluble molecules than bilayers below the phase transition. So having a nice fluid layer here below a very tight layer here is really important. And neutron reflection has certainly given us lots of information about this, and we've got data on this layer as well. So we're building up a picture of that. So hopefully I've shown you that neutron reflection can give us an idea of the structure of an outer membrane model which is relevant to antibiotic action. Finally, I'd just like to highlight the point that the proteins in the outer membrane give rise to very, very repetitive structures, and these are held together by lipopolysaccharide. And we've shown this by solving the structure of these proteins with the lipopolysaccharide molecules, as I show you around here, bound, and we can now see the exact sites where these are bound. So these areas of molecular sieve have very, very important interactions between the porins, these sieve proteins, and the lipids. And it's by this tight interaction that we get this molecular sieve characteristic. So just as a proposal for the future, we need to be able to look at the lateral organization in these, which our neutron reflections cannot do at the moment. So we need to add this lateral resolution, be it GSANs or other techniques, which I'm totally unaware of. But that's the direction which things need to go, I think, for our future studies of understanding outer membrane organization. So to sum up, these are what I believe that we've shown so far with neutrons and what we would need to look at in the future. And I just finally like to thank the people that did the work. My neutron work was kicked off by working with Stephen Holt years ago when he was still at ISIS. That was carried on by great collaboration with Luke Clifton and everybody else on that side. And I've been blessed with having some great people working in the lab as well. So I thank them. And I thank you for listening as well this morning. Thank you.
Thank you very much, Jeremy. Very, very interesting. We have time for a couple of questions before moving on to the second talk. Maybe I can ask Jeremy. The mode of action of polymyxin seems quite non-specific. So is it more chemistry than biology?
Where's the difference? I'm a biochemist, so there is no difference. Yeah, I mean, it's all, I mean, this is it. This is getting down to the level where we, individual bonds is the difference between life and death in people. You know, you can see this in the way that we can modify the structure of antibiotics to get them to outwit resistance enzymes by simply changing the structure of the antibiotic a little bit. So essentially, the lipid polysaccharide, the polymyxin, has found a way to imitate this tight interaction, which is normally the bridging of the calcium and magnesium ions. So it kind of imitates the calcium and magnesium ions. But what's quite clear is that but it also has a hydrophobic tail. It has an acyl chain. And if you take that off, then the polymyxin is several orders of magnitude less effective. And essentially, for that hydrophobic tail to be functional, then the membrane needs to be above this phase transition. So the two things have to go hand in hand. If we knock either of those things out, then if we reduce the charge on the polymyxin, then it doesn't have an effect. And if we get rid of the hydrophobicity. So yeah, it's a molecule that's developed the right balance of charge interactions and hydrophobicity in order to get through this barrier.
Okay. We have a question from Adrian Rennie, who's asking, I thought from your description, is fever part of the response that enables attack on bacteria by natural antibodies?
Oh, I see, the temperature. Yeah, I mean, that could, there's all sorts of theories as to how fever helps us. Of course, we have a fever for all kinds of insults. It's a generalized response to lots of things, including viral infections. But yeah, it could actually help things like this. And there are other molecules, and I think Martin's going to be talking about more molecules which act upon membranes. And we don't really know the importance of phase behavior and lipid domains on the activity of these things. So a couple of degrees, yeah, might save us. So get in that, well, you know, get in the hot sauna as you do in Scandinavia and cure the cold. Yeah.
Fantastic. Okay, we've got a question from Hannah. I think perhaps if we save that for after the second talk, because I can't see that at the moment. Thank you, Jeremy. If you stick around for the question and answers at the end, please.
Okay, I shall stop sharing.
Okay. Second speaker today is Martin Malmsten. He's Professor of Biopharmaceuticals, Biophysics and Drug Delivery at the University of Copenhagen. In addition, he's the director of the Leo Foundation Center for Cutaneous Drug Delivery. Previously, Martin was at Uppsala University as department head. And before that, he was CEO of the YKI Institute for Surface Chemistry. His current research interests include nanomaterials for drug delivery and amphiphilic peptide therapeutics. So the title of Martin's talk is, as you can see there, Antimicrobial Peptides from Membrane Interactions to Drug Delivery. Over to you, Martin.
Thank you, John. Can you hear me now?
Yes, we can hear you.
And also see the shared presentation.
Yes, we can see.
All right, I get going. Thanks so much for the invitation to come and discuss and present some of the work we have been doing on antimicrobial peptides. What I thought I'd do today is to give a little bit of a brief overview of a few different aspects that we have been looking at with regards to these antimicrobial peptides, some of which involving neutrons, but certainly not all, and then end up with a couple of research issues where we are looking forward in our own work and where we think that neutrons for sure could be a valuable and important contribution. All right. So pretty much as Jeremy described, we're starting from the same perspective of the medical need. There is the increased resistance development towards conventional antibiotics has been occurring for several decades by now, but certainly it's not improving, and we see a whole range of both gram-negatives and gram-positives being developed, and so at the same time, the number of new antibiotics coming out to the market is certainly decreasing. Also, a number of antibiotics have been withdrawn from the market for different reasons over the last couple of decades. So there's certainly a need for novel chemical entities to try to address this issue, and we and many others have looked at one class of substances called antimicrobial peptides, which basically form a very central part of the innate immune system. The less famous cousin of the immune system, so to say, that doesn't require antibodies. And to see whether those could be useful at all for therapeutic uses. So for those of you who haven't worked with those or heard of them, the antimicrobial peptides, or also called the host defense peptides, they are a broad class of molecules, thousands of them known by now, but they are of the order of a few dozen amino acid long. Most of them are polycationic. They tend to be quite amphiphilic with a large fraction of hydrophobic amino acids. And basically what they do is that they sit on internal and external surfaces and constitute the first line of defense. They actually do a number of different things, but one of the things they do, sort of analogous to what Jeremy described or showed in that nice video, is that they lyse membranes of bacteria. So that means that it's essentially non-metabolic. It also means fast action. It also, for good and bad, generally means broad spectrum. So we have been looking into questions a little bit on the mode of action and selectivity quite a bit, as well as the other effects of those kind of peptides, not only the killing of the bacteria itself, but also the consequences it might have, for instance, for suppression, inflammation. So over the last few years, we have been working on different areas or concepts. We have spent a fair amount of time looking at identifying new peptides with favorable selectivity, potency and selectivity for bacteria, but low toxicity at the same time. Mode of action, both regarding the antimicrobial and the anti-inflammatory effects. We spent quite some time on a concept which is basically where we just add short tags, we call them, of aromatic amino acids like tryptophan or phenylalanine at the end to boost these kind of different activities. We have translated a couple of those peptides into clinical trials, and recently we are focusing much more on delivery of these kind of peptides. So I will give a couple of examples and illustrations of this. So the first of this is regarding the boosting of antimicrobial effect through tryptophan tagging of peptides. And this is some work that goes back almost 10 years by now. This was one of the first things we did. So what we have here is a very simple, almost prototypic kind of peptide, arginine rich, and it has this sort of 4 tryptophans at the end. And what we did here was a very simple experiment. We took whole blood, and we basically then added to that blood either Staph aureus or Pseudomonas aeruginosa, the 1 gram positive and 1 gram negative. And then we asked the question, can we add these peptides to selectively kill bacteria in the blood without causing any blood lysis? And that's pretty much what's shown here. In the upper panel, we show the hemolysis, and this is the guy in the box, the GR10 with the tryptophans. That is the one we're interested in. We're comparing it to the non-tagged variant and also to 2 best-in-class peptides. At this time, these were the best peptides that were around, so let's say the L37 and the Omeganin, which was in phase 3 clinical trials at that point. And what we see is that we can go high up in concentration, 120 micromolars, and we're still at the negative control level with regards to hemolysis. But at the same time, we do manage to get really nice lysis of the Pseudomonas in particular. Slightly poorer with the Staph aureus, but in both cases better than the best in class. Equally good or better than the best in class. These things, as John asked, actually this is a whole field of research on its own. The specifics of why this specificity comes around. I won't really go into too much of that detail today, but I just rather want to say that you can understand some of this on the first level of understanding by just looking at the different architectures and the lipid composition of gram-positive, so of bacteria versus eukaryotic cells. And of course, there is a vast variation in lipid composition between bacterial strains. But overall, the presence of anionic species and the absence of sterols means that you will have the combined effect will be the increased binding of peptide to bilayers, here observed with a supported bilayer, or also I should say, and facilitated insertion in the absence of cholesterol in particular, which you can see from monolayer studies, and of course the combined effect, the vesicle leakage experiments, which in this case is dramatically different for these kind of peptides, where bacteria mimicking vesicles are very easily lysed, whereas the cholesterol-containing PC-rich lipids are very robust, so to say. I won't go into the details of the mechanisms here, other than to say that from what we have seen with various methods, we don't really see any solid proof of any stave barrels or ordered conformation or anything like that. These are disordered peptides. They are binding preferentially to the outer leaflet, and any defects that we see tend to be of smaller scale, so to say. So there is some nice neutron work that is being done in the last few years, looking at also the effects of peptides on, let's say, flip-flop rates and dynamics, and certainly our indications are more towards that end of local defect formation and maybe of kinetic nature even. But at that point, our interest took us more towards developing these things, and of course the left-hand table is not for reading. The important thing is really the title, that we have tested this against a large number of multi-resistant strains. And of course, since the peptides don't interfere they don't depend on metabolism for their activity. It's rather based on the surface activity. Then it doesn't matter for them whether the bug is resistant to an antibiotic or not. And basically, it means that the peptide is efficient against a number of resistant strains. We have taken one of these peptides to phase 1/2A clinical trials, 2 of them, in fact, and now that particular product is last time I heard it was in the US for continued clinical investigation. The tagging can actually be used also to promote other things, other effects, and what we show here is that by taking a peptide, in this case it's a KY21 from heparin cofactor 2, and you add a little tryptophan tag to that, and you see that binding also to this LPS that Jeremy talked about, this outer lipopolysaccharide in gram-negatives. You see that binding increase substantially, also the binding to the lipid A part of it. And since these guys cause inflammation, they activate cells to generate something called NF-kappa beta, and then various cytokines. And what we show in the bottom part is really that the tagging also then suppresses this let's say inflammatory response by the LPS. In fact, there's also other things that we have worked in relating to targeting of cancer and so on. But what I wanted to show here is really that the antimicrobial peptides have a wider spectrum of activities. Killing of bacteria is one, but suppression of inflammation is another one. This particular one is even worse or even more poorly understood than the antimicrobial effect in itself. All right, but I wanted to shift gear here a little bit, and that is to talk about the need for delivery systems for antimicrobial peptides. And the reason for this is that these guys are big molecules. They are, typically, 15 to 20 to 30 amino acid long, a few thousand molecular weight. They are net positively charged. They are, highly amphiphilic, so they're going to bind to pretty much everything. And that means that both bioavailability, localization of the drug where it ends up, but also other effects are not easy. And so we feel that this is an under-investigated area within that field of delivery systems. So we've spent the last couple of years looking at that, and we work with quite a spectrum of different delivery systems, ranging from the very simplistic conjugations, PEG conjugations, to the more fancy, more academic type of systems. So I'll give a couple of examples of that, starting with microgels. So microgels are basically weakly cross-linked polymer particles. So since they are weakly cross-linked, they are also highly responsive, and that means that they can swell and deswell quite dramatically. In this particular example, we worked with microgels formed with anionic methacrylic acid and ethyl acrylate with a diacrylate cross-linker. Those are basically 150 or so nanometer diameter particles when they're highly swollen. If you load them with antimicrobial peptide, here we have used these two guys on top, LA37 and this N-tag1. We see that we get about 1,000 peptide molecules per particle loaded. There's massive deswelling. They go from 150 or so to 50 nanometer. And peptide loading basically occurs from the core and outward, which means that the peptide, the 1,000 or so molecules that are loaded, they are sitting primarily in the core of those gels. That means for a peptide that is susceptible to proteolytic degradation, for instance, LA37, you can then, of course, as long as it's incorporated into the gel, be protected against bacterial protease degradation, for instance. Also, in this particular peptide, LA37 has some toxicity. Again, you suppress that effect by the loading into the gels or microgels. The antimicrobial effect still remains. So here's some work we did more recently on neutron reflectivity, trying to see, okay, how do these guys, the microgel loaded, the peptide loaded microgels, how do they behave compared to the free peptide, so to say? And essentially what we have done is then basically to take two concentrations of free peptide, either 0.3 micromolars or 5 micromolars, and then we have taken the corresponding basically concentrations loaded then in one of these methacrylic acid microgels. And essentially what we see is that the gels themselves, they do not interact with the membrane under any circumstances at any loading. So that means that basically so it means that it essentially is just the release. So the antimicrobial or membrane disturbing effect in this case requires release of the peptide. Once the peptide is in solution, whether it is basically added as such or whether it's sort of released from gels, in both cases at low concentration, we see insertion in the outer leaflet of the membrane. We don't see any water coming in at that point. If we go higher in concentration, we see that basically spans the entire membrane. We also get a lot of water in there. The main effect of the gel is then essentially one of a container, and the only thing it essentially does is that it slows down the release, which is, you might or might not want to have that, so to say. But this basically illustrates in short that these gels are passive containers. They don't really do anything else than just carries the peptide and then releases it, and that's it. And we have also worked with putting these kind of gels on surfaces and stuff like that. But again, they're basically then just containers. So we have shifted in the last year or two to particles or carriers that are doing something more, if you like. And starting with nanoclays, we wanted to investigate if we could use those for aggregating bacteria for confinement of infection. And this actually started with the work we did together with Arthur Schmidtchen's group in Lund on thrombin, and they have been working on thrombin for a long time, and thrombin, when exposed to bacterial proteases, is degraded. And one of the key proteins or peptides that are being generated is a 96 amino acid guy, which is in the upper right-hand corner. This particular protein binds like crazy to LPS. In doing so, it also formed these sort of beta sheet kind structures. So it's some sort of amyloid formation. It basically forms these lumps, if you like. The same thing happens with if you take gram-negative E. coli with the LPS sitting on the outside. So and what was demonstrated in this paper was that this happens also in wound fluids. So it is an important, we think, defense mechanisms whereby infection is basically confined and prevented from spreading or disseminating into that systemic uncontrolled response that could eventually lead to sepsis. But of course, the 96 amino acid protein, that's not a particularly druggable thing necessarily. It will restrict the number of indications you can target. So we were thinking it could do something simpler, if with the purpose of addressing something like a burn wound or a local cut or something like that in a finger or something. And because of course, as you all know, the clays are quite efficient as aggregating species. And I thought, well, maybe you can use this then for sort of co-aggregating with LPS and with bacteria. And so this is something we published a paper on last year. And on this graph, you have a couple of experiments from that. So here is what we have done here is confocal microscopy. It's combined with something called a live dead assay, which is very simple-minded. I do apologize for that, Jeremy. But it gives an indication of the intactness of the membrane of the bacteria. So if it's green, then basically the membrane is, simplistically speaking, intact. If it's red, then there are defects in it. And what we have here is basically on the left, we have the E. coli that are the control. So they are dispersed and they are all green. If we add the laponite, which is anionic nanoclay, we see that they all lump together. Quantify that in that lower left-hand graph, we see that about 5 ppm or so is enough to essentially have all the bacteria in aggregates. But they're still green. Comparing that to LA37, this antimicrobial peptide alone, what it does is essentially that it induces this defect, as we talked about, in the membrane, but it doesn't cause an aggregation. So we thought maybe we can combine those things. So we both sort of disintegrate them, but at the same time keep them lumped together by essentially loading then LA37 onto laponite. And in fact, that is what we see. In this particular case, LA37 is binding. For the purpose of this experiment, is binding irreversibly. It's sitting primarily on the outside of the laponite, and basically then you have the combined effect of the LA37 and the laponite. What we did then was to see, okay, just what consequences does this have for cell activation? So here we use a reporter cell that you can then trigger by activate by LPS. Then it generates something called NF-kappa B, and which is the mother of all cytokines, and you don't want that stuff essentially. But when you co-aggregate with laponite, you have essentially no LPS free, and as a result of that, we see that both the NF-kappa B activation goes down in a dose-dependent way. At the same time, we don't really see any toxicity to the cells in terms of LDH release. We showed in a following paper that actually this happens largely independent in this range of particle size and set of potential of the nanoclays. I see you're there, John. I'll be a couple of minutes more, but then I'll be done. So something we've been interested in very recently is the effect of topology of particles. And here, this is some work we do together with Dongyang Zhao at Fudan. And he makes this. So we had been working with mesoporous silica particles for some years ago. But now what we have the possibility is to have essentially the same particles, one having spikes and the other not having spikes. And we were interested to see, can we see any puncturing effects of these particles? And if so, can we see any combined effect with the antimicrobial peptides? And to make a rather long story short, we see that we can, and that's what we see over here with this leakage experiments for the or liposome leakage experiments for the bare particles. If we load them with L37, we see much more dramatic effects, both in terms of composition and in terms of structure. I'd be happy to discuss this in detail. Correspondingly, we see that the virus-like particles are much more efficient when loaded with L37 in rupturing E. coli bacteria. All right, so final slide. Where do we see things going now for us? I did talk a little bit about the peptide LPS coaggregation. We think there is a lot more to do there. There has been some neutron work. Jane Lawrence and Katrina Edwards published a nice paper on different small angle neutron scattering work on the kind of aggregates one can get. But I think there is much more room to do more here. These are structures that are very prone to kinetic arrest. And one also now need to look at the consequences for the anti-inflammatory effects. So we are going there for sure. We are going into also the synergisms in mixtures of antimicrobial peptides. We have published on that before using NMR. It's a wonderful technique to show that a synergism between two peptides could be due to, for instance, in this case, a dimer formation with a hydrophobic hub. But it's backbreaking work, and I think larger scale information, for instance, from small angle neutron scattering could be very useful there. We are also investing heavily in photocatalytic degradation. We published a couple of papers there very recently on bare particles, but now we are interested in see what does that do together with AMPs and with the topology. And here we have beam times lined up, hoping to get back to experiments as soon as possible. Finally, since I am, as John said, in charge of a center for cutaneous drug delivery, and this is actually an area where I think AMPs could be particularly promising. We are also moving in towards the issue of skin permeation, where, of course, exchange processes occur and ideally suited for NMR, and possibly where tomography could also be interesting combination. So with that, I'm finished. Thanks for attention. The stuff that you saw is basically from some previous Lotta, Alina, and Randy from Uppsala, a couple of co-workers from Copenhagen, Sarah, Kate, and Eliza, and some collaborating groups and funding at the bottom left. So thank you.
Thank you very much, Martin. We have a couple of questions. I'll perhaps take one which is specifically for you from Paige Stevenson. I know amyloid beta plaques form in Alzheimer's. So does this have any correlation with amyloid aggregating LPS together for infection containment?
That's a really good question. So there are multiple layers on that, but let me put it like this. So the aggregation itself, the amyloid formation, has been shown to have consequences for the antimicrobial effects of antimicrobial peptides. Conversely, amyloid beta, for instance, is known to have antimicrobial effects. The co-aggregation with LPS there is also scattered studies showing that this might have consequences in various pathologies, including Alzheimer, but also in cardiovascular disease. So I think this is a wide open field, and that's why one of the reasons why we're so keen to move into that. We feel that there is lots to be, there are lots of medical and biochemical data popping up, increasingly showing the importance of the coaggregation and amyloid formation. But nobody really knows. There's no distinct answer, yes or no, on this at this point.
Okay, thank you. I've got a question from Hannah Vacklin next. Perhaps, Hannah, you'd like to ask that live? Can you unmute?
Yeah, sure. It's kind of to both of you who spoke. It's clear that there are lots of different kinds of antibiotics and antimicrobials, both old ones that are in use that we don't really understand how they could be made to work better, and new ones. And it's clear that studying all of them and these delivery systems with neutrons is quite challenging. So how do you think that we could best use neutrons to enable the development of actual treatments for patients, given the way we work with neutrons?
Want to start, Jeremy?
Yeah, thank you. So, I mean, I think Martin's a lot closer to this than I am, but from my point of view, there's just, as you say, I think from a more basic biochemistry point of view, how we can help is try to use neutrons with their unique kind of abilities to answer fundamental questions as to why certain antibiotics work in different ways that we haven't understood so far, and then that might maybe help the people that are developing new ones. I mean, some of the models that we use have been used by the Australian groups with Jan Liu and others to look at modified polymyxins and colistins. There's a problem of, so polymyxin and colistin that we're using that I showed you before. So polymyxin I spoke to you about. That's actually used in the clinic these days because there are people who are suffering from bacteria with multiple antibiotic resistance, which it doesn't. And so in spite of the fact that colistin and polymyxins are a bit toxic, you know, it's better than the alternative. And so there's now a resistance building up against colistin and polymyxin, and this involves LPS modification. And so those groups have modified these types of lipopeptide antibiotics in order to overcome this resistance. And then they've studied those in various ways, but also by neutron reflection. And they have a model, and the results from neutron reflection seem to bear that out. So both in terms of basic things and also in targeting specific measurements that cannot be done in any other way. But that has to be the way, because neutron experiments are by their very nature slower and more expensive than almost everything else you do.
Yeah, I guess it was partly about whether and what can we do something to facilitate the process of going from the basic biochemical studies to making links to the people who use those things clinically or want to develop new treatments.
Yeah. So you want to continue, Jeremy, or should I?
No, you carry on.
So I think this is a very it's a very relevant question. I you know, ultimately there are different answers to that. Obviously, the neutron shouldn't be used for anything where it's not providing specific information that cannot be obtained by other methods. Partly because of a shortage of availability, but also because there are quite a few restrictions in the experiments, so to say, in what you can do and so on. But I think that, so for us, we are very translational. We aim to be translational oriented, and so we have identified delivery as one of those areas which we feel is underrepresented. But of course, new targets is equally important. So I think it's difficult to pinpoint from a neutron source facility perspective that some areas should be prioritized and some not. I think if the perspective is translation, I think to ensure in the selection of projects to be accessed to the facility, but I guess also in the interaction itself to make sure that there is the perspective of translation, and so that it's not just or at least to find a balance of projects, some which are more directional and some that are very much working on the very specific things. So it's a hard one.
Yeah, I know. I think often the challenge really is communication and attracting all the right people to participate in those activities that are ongoing at many facilities. Thanks. I think there's some other question, John.
Yes, there's a question from Yuri Kadukov, and I think I know the answer to this is yes, but can you say some more words about virus-like particles? Can they be used to mimic model COVID particles?
I don't know. We started this before COVID, but it is interesting that this sort of local curvature that you have, and how does that propagate to a sort of a local disturbance. I think that, of course, in the COVID bacteria, you have the whole surface composition is fundamentally different. I mean, we have silica essentially, so you have to coat them with something lipid-like and so on, and which may or may not change the curvature and fill in the gaps and so on. But we think that it is interesting, the fundamental questions of topology and how that interplays with other aspects of membrane destabilization could be important, could have consequences for viral infections, although that's not where we have been thinking so far, but looking forward, possibly.
Jeremy, do you have anything to say on that?
No, that's not my field of expertise at all. And I wish you luck in doing it.
I know there's some work at Oxford at the moment on not very virus-like particles, inactivated viruses, in other words. Perhaps I could ask Martin, the peptides, to what extent can we design peptides now for particular functions?
With regards to select, let's say, if the question is with regards to antimicrobial effect and low cell toxicity, I think that the knowledge is relatively good. It still comes with a fundamental limit, I think, that you will not get any very potent antimicrobial effect against very challenging pathogens well below a micromolar. So which in itself limits the type of indications that you could choose. I think that the peptides themselves are the biggest limitation in terms of what we can design or not is really where we put them. So I think we will not be able to target, for instance, meningitis, other than if we drill a hole in the skull and inject it that way, but it's not going to work. So one has to be cautious in where it actually has a good chance of working. In terms of the anti-inflammatory effects, I think that is the field where it's moving the fastest now, and in fact, the health economic drivers for let's say attracting investments into that area and so on is much stronger, so to say. And of course, there's no drug against sepsis still. In that area, the design possibilities, I would say, are slim at the moment. There is room for much more. And that's why I think these sort of co-aggregates between LPS and peptide and how they look and how we can control the kinetic arrest and how the resulting structures actually trigger this sort of activation of cells. That is an unresearched area so far, I say, important one.
Okay. If I may ask another question, the use of nanoparticles as a delivery vehicle, is there a problem getting that from the laboratory to a therapeutic treatment?
Yeah, for sure. I'm sure you have, many of you have seen the discussion in cancer, in the cancer area. Why is nanomedicine not delivering after decades of promise? And again, I think it's partly unfair. There are drugs coming out, and it's partly correct. I think that, again, one has to choose very carefully the indication. So, for instance, the example I gave with the nanoclays and the coaggregation. Let's say if you would take that and inject into the bloodstream, or a very open, deep wound with lots of blood perfusion going on. I mean, you would get emboly formation, and that would be fundamentally bad. That would never get to the clinic. Whereas if you choose a local cut or a burn wound where you have some fluid but not massive bleeding, that's a good indication where there's plenty of room for translation into the market. So it really comes down to choosing the indication and what kind of benefit you can count on in relation to the risk, and as well as the extra investment that is needed to ensure colloidal stability, for instance, throughout the process, both in production and also before and after administration, if that is what you're aiming for. So it comes down to the choice of systems and indications.
Yeah, I know in the area of personal care products, there's some pretty strict regulations about what particle sizes you can use.
Exactly.
I can't see any other questions coming in at the moment. No, if anybody wants to ask anything, now's your opportunity. Okay, it just remains for me to thank both of our speakers this morning then. Thank you very much. Two very interesting talks. Many thanks. So it just remains to advertise the next webinar in this series, which is on October the 22nd. Thilo Perling from the ILL is going to give a webinar on neutron diffraction instrumentation for stress determination. And we'll see you all again then. Thank you very much.